Y'all couldn't even hear. We're gonna look at some more mitochondrial videos. Give me one moment. Oh wow. Okay. So we're gonna get into the mitochondria.
You're in medical school, I can guarantee anatomy is going to be the hardest class you take.
To get out of bed, and every time that you've struggled to keep your eyes open while cramming for a test at 1 a.m., you've actually had a lot of energy. I'm not talking about motivation or enthusiasm, I mean real physical energy. We might be used to energy as a big picture concept. Having energy allows us to move our bodies and do work. That's totally true, but cells also need energy to move their bodies, manufacture new proteins, and make chemical reactions happen. And that's the focus of this.
Not what I was looking for.
Mitochondria are organelles found in eukaryotic cells that play an important role in the production of ATP, the universal energy currency used in all cells. Most mitochondrial proteins are transported from the cytosol into mitochondria through specialized protein translocator complexes. Interactions between these complexes bring the outer and inner mitochondrial membranes into close proximity. The successful targeting of many proteins that reside in the intermembrane space requires translocator complexes that cross both the outer and inner membranes. After entering the inner membrane translocator, many of these proteins do not completely cross the membrane, but instead are released into the membrane and diffuse laterally. They then remain embedded in the inner membrane or are cleaved, releasing a portion into the intermembrane space. Very few resident proteins are simply transported across the outer membrane translocator complex. Matrix proteins do not transit through the intermembrane space, but are directly transported from the cytoplasm across the interacting outer and inner membrane translocators. The protein-rich matrix contains enzymes necessary for cellular respiration, a process during which carbon fuel molecules are oxidized and reduced electron carriers are produced. Invaginations of the inner membrane, called cristae, contain 4 large protein complexes that harvest electrons from these carriers. Complex 2 accepts pairs of electrons from succinate and transfers them through a series of redox centers to coenzyme Q. The lipid-soluble coenzyme Q is reduced by electrons harvested from complexes 1 and 2, and then diffuses through the inner membrane, transferring its electrons to complex 3. In complex 3, electrons are transferred through 2 distinct series of redox centers, which allow them to cross the inner membrane one at a time. Finally, electrons are accepted by cytochrome C, which carries them to complex 4, where they are transferred through another series of redox centers to their final acceptor, a molecule of oxygen, that combines with hydrogen ions to form water. Electron transport in complexes 2, 3, and 4 is coupled with the pumping of protons from the matrix to the intermembrane space. The resulting electrochemical gradient across the inner membrane is called the proton motive force. Protons flow back into the matrix through a component of the ATP synthase called F0. The membrane-embedded ring structure of F0 binds protons in the intermembrane space and releases them on the other side of the inner membrane into the matrix. Proton flow drives the rotation of the ring structure, which in turn leads to the rotation of the F0 central shaft. The rotating shaft sequentially contacts the 3 catalytic subunits of the ATP synthase F1 complex, altering the subunit's affinity for ATP and ADP, and catalyzing the synthesis and subsequent release of ATP. Most of the ATP synthesized in the mitochondrial matrix is consumed outside of the mitochondria. But the inner membrane is impermeable to ATP and ADP. Therefore, an ATP-ADP carrier is responsible for the coordinated import of ADP and the export of ATP across the inner membrane. In cells, mitochondria are distributed near sites where ATP and other mitochondrial metabolites are in high demand. Mitochondria move along cytoskeletal structures such as microtubules and can undergo dynamic changes in shape, including both fusion with other mitochondria and division.
Wow.
Perturbations in the dynamic behavior of mitochondria and alterations in mitochondrial membrane permeability are associated with the early stages of programmed cell death.
It just looked like they were getting to the most minuscule point in development, and instead of transforming or alchemicalizing, it just slowed on down and died. And you have to remember, this is connected to Alzheimer's, also diabetes.
Our body is made up of trillions of cells.
Okay, y'all. I need a little more substance for mitochondria. Mitochondrial control of physiological energy. Let me see. I'm not watching all of it.
My name is Michael Graham Espy, and I'm a program director in the NCI Division of Cancer Biology. And it's my pleasure to introduce today's Walls lecture speaker, Navdeep Chandel, or Nav as his friends call him. Nav received his PhD and postdoctoral training at the University of Chicago in cellular physiology with Paul Shoemaker and Craig Thompson. He's currently the David Chugel Distinguished Professor of Medicine at Northwestern University and is an NCI Outstanding Investigator awardee. Nav began his studies on the molecular and cellular basis of oxygen sensing, which naturally led his curiosity to an ever-deepening understanding of mitochondrial metabolism in diverse cell types. Beginning almost 20 years ago to the present day, Nav has developed the concept of mitochondria serving beyond the more traditional viewpoint of biosynthetic and bioenergetic organelles to include a third distinct role as signaling hubs, whereby the physiologic release of reactive oxygen species and metabolites regulate transcription factors, epigenetics, and to control diverse cellular functions. His recent work has focused on the essentiality of mitochondrial metabolic and raw signaling to control stem cell fate, involving control of cancer cell phenotype and adaptive immunity, as you'll see in the presentation today. In addition to being a champion of basic science, Nav has recently leveraged his transdisciplinary understanding of the mitochondria towards a more translational repurposing of the anti-diabetes drug Metformin as a potential cancer therapeutic. So without further ado, I'd like to invite our friend Nav Chandel up to give today's WALS lecture.
Okay. Can you hear me? In the back? Yeah? Okay, great. Thanks again, Michael, and the folks at the NCI as well as the NIH here for this real honor to give the WALS lecture. As you can see,
keep in mind the NIH is also involved with Dr. Fauci and the virus, okay?
from my title, I'm going to talk about mitochondria and how mitochondria we think are essential players in normal physiology and control of homeostasis, as well as how mitochondria can be co-opted in different pathologies. And much of what I'm going to talk to you is about, as the title says, beyond ATP. And hopefully you'll see why I think that. So the most important slide is this one, because the work that I'm going to present today is an unpublished story from Sam Weinberg, who's an MD PhD student in my laboratory. And he's been helped by two other people in the laboratory, Elizabeth Steinert and Manan Mehta. And we're talking about some previous data from Laura Senna, Frank Weinberg, and Lucas Sullivan. I've had some great collaborators over the years, and current ones as well. Ben Singer, Paul Schumacher at Northwestern, Pen Gao, who does our metabolomics, and Carlos Martinez, who's been helping us do some of our RNA sequencing. And Larry Turka, who's well known for his work in T cells, has been giving us some friendly advice and tutoring about T cell biology. So we have a sort of a very simple question in the laboratory, which is why any mammalian cell respires, right? I mean, if you think about it, it's almost an obvious question. You have a mitochondria, it uses oxygen, generates ATP, and you assume that's the only reason why we have mitochondria, right? And here's some cells which are found in sort of the general tumor microenvironment that we're interested in. We started much of our work in tumor cells, but it occurred to us that the tumors have T cells around them, macrophages, endothelial cells. And then also we started to think, what about other perhaps proliferating cells, right? They're like stem cells. What is the metabolism of normal cells? And this is sort of a question that we've been interested in. Now, just to get you all caught up on the simple biochemistry of mitochondria, as many of you know, there's a respiratory chain. And the respiratory chain dumps electrons finally to molecular oxygen, and this is coupled to a proton motive force, which generates ATP. I think everybody's comfortable with this idea. It's true. It still holds true. But lots of cells do this, right? And the second thing, which was actually the original function of mitochondria, well before oxidative phosphorylation and was worked out, is that the Tca cycle metabolites, and the Tca cycle is coupled to the respiratory chain because the Tca cycle uses NAD and FAD, and they become FADH and NADH. And the electron transport chain of the respiratory chain will make NADH back to NAD so it can keep the cycle going. And these metabolites of the Tca cycle are important for macromolecule synthesis. If you just go back to the great biochemistry books like Leninger and other books, you just follow the carbons. You know, how do you get to a nucleotide? How do you get to a lipid? How do you get to proteins? Amino acids, much of the backbones come from, in part from the cycle. And historically, these are the two functions that have been ascribed to mitochondria. They're there to provide you energy and to give you metabolites for growth. However, we've argued there's a third function. It seems obvious today, but when we first thought about this more than 20 years ago, we had a little bit of a pushback. And the reason was the way many of us were taught biology and where mitochondria sits within a cell is the following. If you have a physiologic signal, right? If, for example, a T cell have to respond to an antigen, or you have to respond to growth factors as to proliferate or to cellular differentiation. Any of these physiological processes at the cellular molecular level, we're sort of hardwired to think those signals are integrated by signal transduction mechanisms, eventually funnel into gene transcriptional networks, as well as remodeling of the chromatin, and then that cell will do whatever it's supposed to do, whether proliferate, differentiate, it makes cytokines, etc. Right? And in that model, once all of this action has happened at the signaling level and the gene transcription, chromatin remodeling, then it just feeds back on the mitochondria and says, I'm going to double myself, proliferate, or I'm going to differentiate, or I'm going to make all these cytokines. Give me whatever energy or biosynthesis capacity I need. And this is almost in a way sort of a passive player in this process, just responding to downstream signaling and gene expression or chromatin remodeling, right? And for us, it didn't quite make sense because this would be like getting in your car every morning and not knowing how much fuel you have, right? And so one simple thing that we thought of is that perhaps mitochondria are not so passive, but they're actually part of that active decision-making process as to determine cell fate and cell function. A cartoonistic way of thinking about this is if you have those signals, yes, they activate many of the same signal transduction pathways and try to remodel chromatin and genes, but they have to go through an obligatory step, which is to assess the mitochondrial fitness. In other words, signals that have to communicate from the mitochondria onto the nucleus, right? And what would these signals be? Now, we got inspired by two findings. When we first reported this paper on hypoxia control of the HIFs, which is the dominant transcription factor in hypoxia, what this signal could be. So when Jiadeng Wang and colleagues found cytochrome C release that happened under cell death, we started to think, gee, mitochondria must be releasing things. Of course, that signal was a death signal. And we thought, well, mitochondria obviously are also selected for a positive signal, a beneficial signal. Well, one obvious one was reactive oxygen species. And Sugo Reed, who's luckily here back from Korea, was one of the pioneers who proposed that and did some of the early work showing that reactive oxygen species could be a signaling molecule. And I think that biology continues to expand. And Sugori and Torn Finkel and others who were working on this had proposed NADPH oxidase as being one of the critical generators of ROS. And we thought, well, maybe the mitochondria could also be that generator of ROS. People didn't like that idea because, in their simplistic notion, mitochondria only generated ROS when they were damaged. And this explained neurodegeneration. Maybe this explained heart failure, or the free radical theory of aging, and this whole idea that mitochondria are bad actors. And so one of the things we continued to show over the years was that that's not quite true, right? In many instances, we think mitochondria do release reactive oxygen species under physiological stimuli to control a variety of important transcriptional nodes, including Notch signaling, HIF, and NF-kappa B. And so the respiratory chain isn't perfect, and the electrons from this chain can leak out to make superoxide and hydrogen peroxide to make these mitochondrial reactive oxygen species that are released under a variety of stimuluses is to control these very important transcriptional networks. The other thing was that more than 15 years ago or 20 years ago also, we were the epigenetic and the chromatin fields were discovering all the enzymes over the decades, like the histone acetylases or the lysine demethylases that control chromatin remodeling, right? One thing that was obvious to someone who works on mitochondria is, gee, isn't it interesting? They all use Tca cycle metabolites to work, right? Where does your acetyl-CoA come from, or your alpha-ketoglutarate to make these demethylases work? And things like succinate are powerful inhibitors of potentially of these demethylases. I have quite a wonderful colleagues at Northwestern, including our biochemistry chair, Ali Shladafor, many of you know, for his work in the chromatin field, I joke to him all the time that he's just looking at an output of mitochondria when he studies epigenetics, because all of these things we've been able to link to the Tca cycle metabolites controlling the histone acetylation. So if we restrict the generation of citrate here, we can impact histone acetylation, and we've published on this before. So in thinking about what functionally mitochondria do, I think we can all, many of us are comfortable that the mitochondria might be important for survival by generating ATP and keeping the ATP-ADP ratio high to drive many cellular reactions. Just remember that glycolysis can compensate because it can also generate ATP. The other one is that it's important for biosynthesis. In other words, macromolecule lipids, nucleotides, many of the macromolecule synthesis also comes from mitochondria, so it's a biosynthetic organelle. And then the third function, and this is what my lab's really worked a lot on, is this idea that there's things being released from mitochondria, metabolites or reactive oxygen species, which can then control transcription through either chromatin remodeling or directly by activating transcription factors. And so with that as a background of what we think mitochondria do, the three distinct functions, is it plays out very differently in different cell types in vivo, right? So it seems like the cancer cells use mitochondrial metabolism really for growth and survival, where stem cells, and in particular hematopoietic stem cells that we've worked on, they really don't care about their ability to proliferate. And so if we knock out the same respiratory chain genes in a cancer cell compared to stem cells, it's completely different. The stem cells continue to proliferate. The fetal stem cells, hematopoietic stem cells will proliferate, but they won't differentiate into progenitors. And the adult ones, which tend to be quiescent, lose quiescence and undergo actually hyperproliferation and stem cell exhaustion, right? Completely different than the cancer. And why that is, we're still trying to figure out. And finally, T cells, it seems to be all about their ability to function. So if it's a regulatory T cell, it doesn't suppress. And I'll show you what I mean. So I can't go into all these model systems, but as a simple compare and contrast in vivo, I'll focus a little bit on cancer and a little bit on regulatory T cells to show you an example. So we got into the cancer metabolism field more than about 12, 13 years ago. As you know, that the cancer metabolism field has been revived, in part by an old observation made by Otto Warburg almost 90 years ago now, which he observed that cancer tissue under normal oxygen conditions right on the laboratory bench would make lots of lactate. This is called the aerobic glycolysis or the Warburg effect. Like the clinicians know that using PET imagery, you can see with a glucose tracer PET positive, right? So yes, tumors are glycolytic. And there's been a lot of hype around that to the point where people think that the mitochondria are dispensable in cancer. So we thought, well, that doesn't make sense to us based on our simple biochemistry knowledge of what mitochondria do. And we invested early on in making mouse models, using mouse models of cancer, like the ones that Tyler Jacks and Dave Tuvison and others have pioneered, but also making some models that we could use in vivo to test the necessity of the respiratory chain. So one of the ones that we made was TFAM. So TFAM encodes for, is a nuclear protein that is necessary for mitochondrial DNA replication and transcription. So remember, the mitochondria has its own genome, and it encodes for certain key subunits of the respiratory chain. And so we generate a flox mouse. So wherever you knocked out TFAM, mitochondrial DNA is gone. If mitochondrial DNA is gone, guess what? A variety of the critical subunits of the respiratory chain are gone, and you don't have a mitochondria that respires. Functionally, the at a cell biology level, if you look by electron microscopy, the mitochondria are there, they just don't respire, right? And so we asked what happens in one of these models that the field uses, and the one we used is the lock, stop, lock, skip.
Y'all, this man just said he named people that created different cancers. Hold on.
Gone, guess what? A variety of the critical subunits of the respiratory chain. And we invested early on in making mouse models, using mouse models of cancer, like the ones that Tyler Jacks and Dave Tuvison and others have pioneered, but also making some models that we could use in vivo to test the necessity of the respiratory chain. So one of the ones that we made was TFAM. So TFAM encodes for, is a nuclear protein that's necessary for mitochondrial DNA replication and transcription. So remember, the mitochondria has its own genome, and it encodes for certain key subunits of the respiratory chain. And so we generate a flox mouse. So wherever you knocked out TFAM, mitochondrial DNA is gone. If mitochondrial DNA is gone, guess what? A variety of the critical subunits of the respiratory chain are gone, and you don't have a mitochondria that respires. Functionally, the at a cell biology level, if you look by electron microscopy, the mitochondria are there, they just don't respire, right? And so we asked what happens in one of these mouse models that the field uses, and the one we used is the lock-stop-lox KRAS, oncogenic KRAS-driven lung cancer model. So here we just dump a little adenovirus crete, and the lock-stop LOX allele is gone, and it activates oncogenic KRAS, and you see an increase in tumor burden. To the extent that you can knock out TFAM at the same time and excise the TFAM gene, we notice a lot less tumors. So remember, by knocking out TFAM, we have made the tumors completely dependent on glycolysis, right? The ultimate Warburg cells, as I would call them, but yet you have less tumors. And this simple experiment basically told us the necessity of the respiratory chain, and at least under these mouse models, for tumorigenesis. As Mike alluded to, one of the things we also got into was metformin, because the epidemiologists had noticed that people who had taken metformin for their diabetes had a lower incidence of cancer across a variety of cancers. This has actually led to some clinical trials, including a large-scale one that's happening in Canada in breast cancer, and the results will be out next year. But sometimes when a drug is really cheap and can be easily repurposed, and obviously, unfortunately, there are patients suffering from the terrible disease of cancer, you sort of rush to do the clinical trials. And I'm not completely convinced we've been very, we've been very thoughtful about some of these trials, because it's not clear whether the anti-diabetic dose should be the dose for the anti-cancer. In fact, our studies, at least in the mice, suggest that you need almost a maximal tolerated dose of metformin to have any efficacy. And not every tumor responds, because in order for metformin to get into a tumor cell, it needs the organic cation transporters. In the absence of that transporter, you can throw buckets of millimolar amounts and nothing happens. But the other question was, what's its target? And in 2000, there was 2 reports in JBC that suggested that mitochondria, if you give isolated mitochondria metformin, you can inhibit complex 1. This is complex 1. There's 45 subunits in complex 1. Now, the best way to show what a drug does, I still think is the gold standard, is to find out where it binds in a particular complex or protein, mutate a certain residue so it doesn't change the catalysis of that complex or that protein, but just makes it refractory to binding, right? That would be the gold standard. I'm not a structural biologist. We didn't do an elegant experiment like that. It's a 45 subunit complex. It's pretty big, pretty complex. But we did something a little clever, I think, which is we said, okay, let metformin inhibit mitochondrial complex 1. And if it's necessary for its anti-tumor effects, let's rescue it by putting in the cancer cells a protein called NDI1. NDI1 is found in yeast. Right? It's a Saccharomyces cerevisiae. It does the same thing that complex 1 does, NADH to NAD. The only thing it doesn't do is it doesn't pump hydrogen. And by doing NADH to NAD, it can donate electrons to CoQ and down all the way to molecular oxygen. So it's our functional rescue. So we did this in a colon cancer model. We did this in a lung cancer model. And as you can see, we let the tumors grow. If we give it metformin in the drinking water, we can inhibit tumorigenesis. If there was NDI1 there, they were refractory. They didn't care. Right? They're all isogenic controlled except plus or minus NDI1 or plus or minus metformin. What's nice is that once we published this a couple of years ago, a bunch of other physician scientists and clinician scientists have now gone and given metformin to their patient cohorts and taken biopsies to do metabolomics. And we know a signature when mitochondrial respiratory chain is inhibited. We sort of know a metabolic signature that's consistent with that. And now, giving metformin to those patients, you can see that signature, implying that metformin, even in human patients, can get into their cancer cell mitochondria and perturb metabolism. So one of the things that we've been trying to figure out is exactly what aspects of the respiratory chain are critical. I mean, do you need all this ATP that's coming, or is it just about the metabolites? And one way we've thought about doing this is we've knocked out mitochondrial complex 3. If you knock out complex 3, it's right in the middle, right? These electrons aren't going to flow, and you won't proton pump. ATP is gone. The Tca cycle won't work or canonically like it should, and then maybe you won't grow. And so if you knock out complex 3, consistent with what everything else I've shown you, again, you get very little tumors growing out. Okay. Now again, we've gotten into giving these ancient enzymes to complement these mutations. So here's another one where we take the complex 3 knockouts, that's all gone. And here we put a protein called the alternative oxidase. It's found in plants, it's found in sea squirt, and in many lower organisms. And it can take electrons from Q and again transform to molecular oxygen. Very similar to cytochrome C oxidase. But again, here it'll allow this Tca cycle to occur because both complex 1 and 2 activity is regained because it can donate all its electrons to AOX. And when you do that, compared to the control complex 3 knockouts that don't grow, here by putting back AOX, we can recover tumorigenesis. So in this simple experiment, again, it starts to point that one of the key functions of mitochondria, we think, is to generate those Tca cycle metabolites. It's for growth. And really a model that's emerged from my lab and people like Ralph Debredinas, who's done some nice work in human patients with glucose tracers, as well as work of like Alec Kimmelman and others, has come to, this is sort of a bird's eye view, that in the land of plenty, when nutrients are available, the mitochondria is a biosynthetic bioenergetic hub, a line for rapid cell proliferation, right? It can give you all the lipids, it can help you make nucleotides, it can give you the energy. But as many of you know, tumors sit in harsh environments. And there, many of the pathways that the cancer metabolism community has discovered, and we haven't contributed much to this, which is things like macropinocytosis or autophagy or branched-chain amino acids or fatty acid oxidation, the activation of AMPK, all these survival pathways, one of their jobs is to catabolize whatever they can in a cell to feed the mitochondria, make enough energy so you can at least survive until you get neovascularization and you go back to the land of plenty to grow out. So under both conditions, as a nutrient-depleted condition, it's an anabolic machinery. In a nutrient-depleted, it's a catabolic machinery to generate ATP for survival. And I think this is a model that we continue to test, and obviously we're looking for nodes that we can interfere with for cancer therapy. So really the take home here is that we think the in vivo role of the respiratory chain is control proliferation and survival. I won't have time to show you the stem cell data, but we can do similar genetic tricks, knocking out complex 3 or other ways to knock out the respiratory chain. As I alluded to, the stem cells themselves are there. They're phenotypically, they look like stem cells, but they don't do what they're supposed to do, which is generate progenitors. Right? So they don't have a proliferative defect, unlike the cancer cells in vivo, but they have a differentiation defect. And the T cells, which I'm going to show you, is all about their function. And so we got into this almost 7-8 years ago, a talented MD PhD student, Laura Senna. And again, we were sort of watching the field of immunometabolism that continues to grow and evolve. And again, the historic bias has always been that they're very glycolytic, right? And in fact, rapidly proliferating cells do show robust glycolysis, but that's not should not be misinterpreted that their mitochondria are not equally important. And so again, we do very simple experiments. We ask the question, is the respiratory chain necessary for proliferation or T cell activation? And could we use some of our newer mouse models? So one of the ones we've been using is knocking out the catalytic subunit of complex 3. So complex 3 has 11 subunits. The risky iron sulfur protein is encoded by the nucleus. So again, we can flox these alleles. Let's knock out complex 3, cross it to CD4-CRE, and again, the respiratory chain won't work. If the respiratory chain doesn't work, you don't make ATP, obviously. So these cells become purely glycolytic. Their growth may be compromised because you may not make enough Tca cycle metabolites. It's the ROS that's being generated by complex 3 for signaling might be affected. The metabolites for histone acetylation would be affected. So there's going to be multiple effects, and it gets quite difficult to start discerning all the reasons why. But Laura initially did some simple experiments. One of them is you can take a RAG-deficient mouse, a lymphopenic mouse, and adaptively transfer it to wild type or risk knockout. This is sort of this homeostatic expansion of T cells, a very bread and butter assay of the community. What she noticed was no phenotype. The wild type cells, wild type T cells, you put them in this RAG-deficient mouse, it proliferates fine, as others have shown, and so do our complex 2 knockouts. We suggested that there's no growth defect. I did remind Laura that, the T cells are there to respond to antigens, that perhaps she should try a variety of antigens. And when she did that, she got the opposite response. So they have the ability to grow under these homeostatic conditions. They just don't do it when they have to respond to an antigen. And that started to smell like signaling because they can find a way to grow and survive and under one condition, but not the other. And without going through a lot of the mechanistic details, as this was published a while back, and I want to show you some of our more recent unpublished data, but this is sort of was a nice experiment. So remember, the complex 3 knockouts are purely glycolytic. So you could argue there's something about the fact that they're too glycolytic or something about energy that was a problem. But we, like the signaling paradigm, and one of the ways we think signaling is by generating mitochondrial ROS. So if you look at IL-2 production, this is in cell culture. The wild type cells, when they're stimulated, they make IL-2, the classical cytokine of T cells being activated. The complex 3 knockouts, or risk knockouts, is this marked reduction. And then all we do is we titrate in nanomolar amounts of galactose, of H2O2. And we do it by giving galactose oxidase and galactose. Galactose is a substrate for galactose oxidase, and we can titrate the substrate and the enzyme concentration and to whatever level we want. We can generate nanomolar, micromolar, millimolar amounts of H2O2 continuously. And by just flooding the system with nanomolar amounts of H2O2, we asked whether in our risk knockout, we can bring back the IL-2, and we can, right? So the black bars are the risk knockouts. So they have very little IL-2 when they're stimulated. As long as we provide them only one thing back, H2O2, we can bring back IL-2 levels. And that suggested that one of the things that was important in the absence of complex 3 was perhaps a ROS production from complex. And we showed that using dyes and all sorts of other methods. But the simple mechanism that we outlined was that the early signal, once you activate the T cell by the T cell receptor engagement, is an influx of calcium. That's one of the earliest signals you can detect. And in large part, that signal was intact in our knockouts. What wasn't intact was the generation of hydrogen peroxide in our knockouts, and that was necessary for NFAT translocation into the nucleus for IL-2 production. As many of you know, NFAT is one of the key transcription factors that's necessary for IL-2. NF-kappa B and AP1 are the other two, and they were not affected. So there was some specificity to this pathway. Okay. And so the idea we had is that the calcium would enter the mitochondria, where it would activate the dehydrogenases is to pump up and rev up mitochondrial respiratory chain activity, and a byproduct of that is H2O2, which would then allow for this NFAT-dependent IL-2 production. So when Sam Weinberg, a talented MD PhD who's graduating next month, wanted to follow up and start to ask, beyond activation, what are the different subsets? What is the role of the respiratory chain in a memory cell, or in an effector cell, or in a regulatory T cells? And we like the regulatory T cells because there's a lot of nice literature in regulatory T cell. Was also much of how the Treg regulatory T cell literature, there's a lot of epigenetic control of Treg function, which to us, like I said, we like to think of chromatin remodeling in the epigenetic landscape because we can see how it nicely ties into mitochondrial function, because many of the Tca cycle metabolites control those enzymes. So again, it's a very similar approach. We take the FOXP3 Cree, and these mice also have a YFP, so you can tag them if the Cree is active, and cross it again to the complex 3, the risk flox mice. So you have complex 3 gone again, and what happens? And he got a completely astounding result, which is he got a mouse that looks like a FOXP3 null mouse. So FOXP3 is sort of the lineage-specific transcription factor necessary for regulatory T cell function. If you lose FOXP3 in a mouse, you get the scurfy mouse. In 3 weeks, that mouse dies. There's autoimmunity. There's immune dysregulation everywhere. There's hyperinflammation. And patients that have mutations in FOXP3 also had this sort of hyperinflammation, immune dysregulation. And we got the only mouse that I know that almost phenocopies the scurfy mouse. And I couldn't believe it. And genetics don't lie, but sometimes they do. And so we just made sure if that's true, we should take another subunit of complex 3. There's 11 subunits. One of them is encoded by the mitochondrial DNA. The other 10 are nuclear encoded, and all of those 10 are all necessary for the complex. If you lose any one of them, the complex doesn't work well. And this is the QPC protein, and we got an identical phenotype. So all the data I'm showing you, we sort of phenocopied with each other mouse, right? That was good. So even though we were quite excited by this finding, there was one simple experiment Sam had to do for me to get excited. Like, listen, if you show this to anybody, all they're going to say is you have a dead mouse because you had no Tregs, right? You've got a mouse that the Tregs needed for survival. So tell me what the Treg numbers are. And if they're completely gone or they're really low, it's a cool finding, but it ain't that cool. You need an ATP or something like that, right? And what he found was the opposite. The number of FOXP3 positive cells were identical between in fact, they were a little bit more in the complex in knockout, like they were, right? And you can see if you take out these numbers out and you put Ki67 or CD44, which is an activation marker, phenotypically, we've got plenty of FOXP3 positive so-called Tregs that proliferate. They look like they're activated, they're surviving, they're proliferating. So we're down to the last function, right? Are they suppressive? And here I'm showing you, we've done the in vitro suppression assays and they don't suppress very well. Here's the in vivo. And this is where, again, we go back into the rag mice where we either give a wild type effector cells, and you can see these mice develop colitis and immune dysregulation, and they die. If you give the wild type effectors with the wild type Tregs, right? And you give enough Tregs that should suppress the effector cells, a classic suppression assay, the mice survive, right? But now you redo that experiment where you give the wild type effectors with the knockout Tregs, and again, it looks like they never got any regulatory T cells, right? And this told us that we have Tregs that are there, they can proliferate, they can survive, based on many markers that would be called essential for Tregs. Many of them are there, including CTLA4, FOXP3, but they just don't suppress. And another experiment we can do, now we go to an inducible system. So instead of using the FOXP3 Cree, which comes out in development, this is the FOXP3 tamoxifen inducible Cree. So we let the mice get to adult, and now we add a B16 melanoma, the classic sort of, in my lab, we don't try to reinvent too many of these techniques other than our mitochondrial stuff. You get a nice tumor that grows out. And now we're adding tamoxifen. And remember, the tamoxifen should activate Cree only in the FOXP3 positive cells, the Treg cells. And by knocking out complex 3 inducibly in the regulatory T cells, obviously we've allowed these Tregs to stop suppressing and unleash immunity, and they make very small tumors that eventually get cleared out, right? And so that tells us that complex 3 is necessary for Treg function. The question is why? So here, what we've done is we've taken the Tregs out and shot it through RNA-seq analysis just to get an idea what's up and what's down. The number one pathway we consistently see in vivo when mitochondria are, the respiratory chain is impaired in our stem cells and our T cells, so far is MYC. MYC tends to be up, and so does mTORC1. Those pathways tend to be up. And it's almost as if the system must sense something's wrong and is trying to compensate by activating. And remember, MYC and mTORC1 are the two of the pathways that control metabolism. MYC almost regulates just about every metabolic gene, and mTORC phosphorylates many substrates in the lipogenesis and nucleotide synthesis pathway. So this must be sort of a feedback on the system. But the other thing was what's downregulated, right? We think that it must be downregulating suppressive genes. And its two obvious ones were FOXP3 and CTLA4, but they weren't. But then we started to look into this a little bit more, and here's some genes that have been associated to suppressive regulatory T cell function. And in fact, if you lose any one of these genes, specifically in regulatory T cells, eventually you develop an autoimmune like disorder. Some of these, like neuropilin-1, takes more than a year, some of them less. But remember, we're having a whole host of these genes all coordinatedly downregulated. It's almost like we've made multiple knockouts of all in one. And so how do you coordinate a widespread suppressive gene downregulation? And what are some of these, how do you get all of these genes perhaps to downregulate? And so this is where we turn a little bit to DNA methylation and histone methylation. So there's a large family of enzymes now called the alpha-ketoglutarate dependent dioxygenases. I think my colleague from the hypoxia community and friend Bill Kalin gave the walls a couple of weeks ago, and he was really a pioneer in figuring out one of the early ones, which is the EGL9, the prolyl hydroxylases. So these are the regulators of HIF, right? So they control hydroxylation of HIF, and they're alpha-ketoglutarate dependent. And some of the other ones are the Jumanji domain, the TEP enzymes that participate in DNA demethylation. And so the histone lysine demethylation, the DNA demethylation, all these reactions are driven by alpha-ketoglutarate. They use oxygen, they make CO2, they use iron, and the byproduct is succinate. All you have to do is go to PubMed. Alpha-ketoglutarate is mitochondria. Iron is controlled by mitochondria. Oxygen availability is controlled by mitochondria. Succinate is generated in the mitochondria. You can see how early in evolution, and the one way that mitochondria might have communicated to the nucleus or to the cytoplasm is through these enzymes, because it can potentially control the activity by a variety of ways. One thing that's come out of the people who've studied these enzymes is the most potent regulator is 2-hydroxyglutarate. Succinate will also inhibit this enzyme, but not as effectively as 2-hydroxyglutarate. And remember, 2-AG and alpha-ketoglutarate are the same molecule. One is this oxidized and reduced versions of it, right? The cancer people all know about 2-hydroxyglutarate, right? This is the excitement that's gone in the cancer metabolism field because the IDH1 and 2 mutations that are found in gliomas and AML make 2-hydroxyglutarate. And much of our understanding of what 2-hydroxyglutarate funneling into these enzymes comes from that literature. Now, the type of 2-hydroxyglutarate that the IDH mutations make is the R form or the D form. So remember this chirality. There's another version called the L form, right, or the S form, that every person in this room can make without that IDH mutation. Plants make it. It's been conserved throughout evolution, okay? And I want to talk about how do you make not the IDH mutation version of 2-AG, but the L. Because I think the implication of this is much broader, and I'll tell you why in a second. So the way you make L2AG, or the S2AG, is you only make it when NADH levels are high. Right? And NADH levels are high if the respiratory chain is inhibited or impaired. Right? Remember, complex 1's job is to take NADH, make NAD. So most of us have a lot more NAD than NADH in our mitochondria. But if we're severely hypoxic, or if we take a poison that inhibits our respiratory chain, or as many have alluded, that diseases like Parkinson's or natural aging, you see a downregulation of the respiratory chain, and you start to see a buildup of NADH compared to NAD. And there's a whole literature around giving NAD supplements now. And I've asked people, what does NAD do? And I think this is one explanation. And when the NADH levels increase compared to NAD, malate dehydrogenases. And remember, malate dehydrogenase takes malate usually and converts to oxaloacetate, right? Malate is its natural preferred substrate, but it will promiscuously utilize alpha ketoglutarate. And if there's a lot of NADH around, the malate dehydrogenases will convert this to 2-hydroxyglutarate, right? The key is you need that NADH to drive this reaction. And once you have the 2-AG, there's an enzyme called 2-hydroxyglutarate dehydrogenase that will get rid of the 2-AG buildup. And the brain has the highest levels of 2-hydroxyglutarate dehydrogenase. Patients which have a mutation in that enzyme will slowly accumulate 2-AG, and they get all sorts of neurodevelopmental and neurodegenerative phenotypes. So we can see how 2-AG might be linked to neurodegeneration. One key thing is this enzyme is completely dependent on dumping its electrons to complex. So when we knock out complex 3, and we first did it in our stem cells in vivo, we saw a lot of 2-AG accumulate up to 1 millimolar. And it's being driven because the NADH builds up in our complex 3 knockouts, and you can't get rid of it. The other place is LDH with acidic pH can also generate 2-HG. So highly glycolytic cells, those can also start to accumulate. But again, you need the NADH to drive that. And you know, we've now have many ways to reconstitute with ancient enzymes, replenish the NAD pool in our complex 3 knockouts, and we can bring down the 2-AG. So we're quite confident that this is the mechanism by which you generate the 2-AG, an NAD malate dehydrogenase dependent system. And so again, the key is whether that 2-hydroxyglutarate accumulates in our Tregs, and it turns out that the wild types have almost like 50 to 100 micromolar, and it accumulates up to somewhere between 3 to 400 micromolar. But the key is, how does it translate to alpha Kg, right? Because it has to outcompete the alpha Kg, and we start to see, again, this is doing metabolomics right out of the mouse. If a mouse that's going to die within 3 weeks, right before it, and we just take it out and shoot it through mass spec, these FOXP3. The succinate ratio to alpha Kg also goes up. And so the question is, does this correlate with anything to do with DNA methylation, right? So the TET enzymes participate in DNA methylation, and they've been shown in vitro to be very sensitive both to alpha Kg and the succinate. Right? They're alpha Kg dependent, and they're sensitive to 2-AG and succinate. So if you put in 2-AG or succinate, you can start to see hypermethylation of DNA. And so what we did is we went back to our RNA-seq and said, let's look at the 200 genes that are downregulated, and now do the bisulfite DNA methylation sequencing, and now looking at the top downregulated genes. And if you look at their CpG islands, as you see a hypermethylation compared to the wild type, right? So there is a correlation between the loss of complex 3, the loss of suppressive function, downregulation of key suppressive genes, hypermethylation of the DNA, and increase in things like 2-hydroxyglutarate and succinate. They're all correlative. Now, the reason I keep on saying I'm very careful about this, my original degree was in math, and one thing you learn over and over is causality. Unfortunately, I don't have a good causal experiment, right? The causal experiment would be to take our complex 3 knockouts and find a way to get rid of 2-hydroxyglutarate and fix the phenotype, if that's the driver, right? The best way would be to overexpress this enzyme. We can't, and we've tried. The problem is it's dependent on complex 3 knockouts. If we had a complex 1 knockout, which would allow the NADH to build up and make 2-hydroxyglutarate, we could in that system overexpress 2-hydroxyglutarate because complex 3 is intact when complex 1 is knocked out, right? So we're going back and doing those sort of experiments. But one experiment we can do is sort of an in vitro sufficiency. We can take our FOXP3 null, wild type FOXP3 right out of a mouse, give it all the cytokines, keep them happy, get them to proliferate, and give it 50 micromolar 2-hydroxyglutarate. 50 micromolar gives you about 400 micromolar in the cell. So if you put 50 micromolar in the cell culture, and by metabolomics you ask how much the concentration is enriched, it's almost tenfold. And it's about what we see in our knockouts. So we're on par So in other words, you could always, this is one of Mike's favorite things he likes to say, you can always dump in as much of a metabolite or a toxin until you see an effect. We were careful to put back the amount of hydroxyglutarate that we see in our knockouts to simply ask whether in a wild type cell, whether giving it that amount of 2AG sufficient to drive down regulation of some key genes, including neuropilin and TIGIT, which were the genes that we observed were downregulated. By contrast, FOXP3.
Okay. We're gonna look at a little bit of this, and then I'm gonna go because it's already 56 minutes in. Didn't mean to get on here and stay, but this is so interesting because what he was saying was that they take mice and do these tests, and he did mention that they were creators of different types of cancer, and it just makes you wonder. This is the NIH, and that's what they were talking about, gain of function study. And so trying to make something functional but not detectable. And it just makes me think, I wonder how many of these diseases have they created in a lab. But let's just watch a minute of this and then I'm gonna go.
It's a pleasure to be here at Google to talk about our research and let you know all the great things that we have going on. And thanks for the support that Google Giving provides for organizations like ours. So we are a nonprofit research foundation that does research into aging. And to sort of start you out with our concept of how we think about aging and how it impacts disease. The traditional way that people, that medicine thinks about intervening with aging is attacking the pathology. So you've got a disease, a heart disease or cancer, and you've got a cancer. What is cancer? It's too many cells in a tumor. So you got to get rid of the tumor. So we're gonna zap they're gonna cut out the tumor, zap it somehow. That's a pathology approach to dealing with a disease of aging. It's been kind of hip lately to go back to the beginning and look at metabolism. So the root cause of all aging is the byproducts of metabolism. And so there's all kinds of cool genetic tricks you can do in animals, in model systems, where you can genetically manipulate them and slow down their aging, 10, 20, even 100% in some model systems. And maybe you'll get a doubling of the lifespan of a microscopic worm or a fruit fly, and that works on up into mice. But people are looking at trying to intervene at that level and saying, is there a way that we can drug some of these pathways and see if we can slow down the aging process a little bit? And that's a fine approach, but our preferred method is to intervene at the level of where the damage is accumulating as a result of metabolism, as a way to treat pathology by either reversing or preventing damage that causes the pathology. To step back for a moment and talk about what our foundation does in the big picture, we organize outreach events. We do some interaction trying to get policy changes, say at the FDA and such like that. And most of that is focused in on different conferences that we organize. The last 3 years, we've been organizing a series called the Rejuvenation Biotechnology conference here in the Bay Area, which is focused on trying to nucleate a rejuvenation biotechnology community around companies and academia, nonprofit research, trying to build this industry. Another aspect of our work, non-research work, is our educational mission. The main aspect of our educational mission is our summer interns who are focused on biology, biomedical research, and bring them into our labs here in Mountain View, or distributing around the country to affiliated labs where they'll work on a project that has some interaction with our mission. World-class scientists on our research advisory board, people who we will convene, main funders of biological research, certainly biomedical research in this country, and they spend a bit over $30 billion a year on research. The National Institute on Aging is a piece of that NIH pie, and their budget's a little over $1 billion.
Wow.
Their actual expenditures on stuff that we consider relevant in this area. So we have in our research aspect, which is the main aspect of what we do at SRF, we have research at our successful in recent years, and our researcher David Spiegel is a professor at Yale that we have been funding on this project. It was known that a certain kind of glucose crosslink. So glucose is a molecule that everybody here is familiar with, right? It's a unit of energy that our body uses to transport energy through the bloodstream. It's also a fairly reactive molecule, and so it can react with, say, the walls of your arteries and cause them to stiffen up with age, because we always have, whether or not you have diabetes, which is just a little bit too much glucose floating around in your bloodstream, or the normal amount, you still have glucose floating around in your bloodstream, and it's going to react with anything that it touches. Slowly, over time, those reactions build up, and eventually you get stiffening of the arteries, you get diabetic neuropathy, and associated effects of aging along those lines. So what David was able to do was synthesize one of the main chemical crosslinks that form synthetically in the lab, and now we're working at figuring out ways to break those crosslinks to both diagnose, detect them in the body, and break them. And another mission of ours, like I was saying before, is to try to stimulate an industry here. And so here are a few companies that have spun out of our technologies that we've developed at SENS Research Foundation. ICOR, where they are working on therapy for macular degeneration based on some enzyme therapy research that we did in our lab. Ocean, which it.
All right, y'all. We're gonna have to catch this one on another day. We're gonna remember to start it off at like 45 minutes to get the introduction and stuff. But this is another organization that does research. Google. He's speaking at Google, and a lot of their funding comes from NIH. So that is it for the night. Peace and power. And I'm out of here.